Dear students, today we will talk about genetic crosses. Genetic crosses can be defined on the basis of character and nature. On the basis of character and nature, we define the genetic crosses. So genetic crosses can be of different types on the basis of character and on the basis of nature. First, we will talk about the various types of genetic crosses on the basis of character. On the basis of character. On the basis of character, genetic crosses may be monohybrid, it can be dihybrid, trihybrid, and so on and so forth. As you know that every character has two different forms, has two contrasting traits. Each character has two different forms which are known as contrasting traits, and every trait is encoded by two alleles. On the basis of character, firstly we'll study about monohybrid cross. Monohybrid cross. What is monohybrid cross? Monohybrid cross is the cross in which we study the inheritance of only one character. It involves the study of inheritance of one character at a time. It means that at a particular time we study one character. In monohybrid cross, there is an inheritance of one character. Here a term is inheritance. What do you mean by inheritance? Inheritance means the transmission of characters from parent to offsprings. In this cross, we study how the characters are transferred from parent to their offsprings. And this transmission of characters from one generation to next or from parent to offspring is known as inheritance. And in monohybrid cross, we study the inheritance of how many characters? Inheritance of one characters. Okay? For example, height of a plant. Height of a plant. This is the one character. As you know that every character has two different forms. Suppose I am moving along the roadside. I see so many plants. Some are very large and some are very short. When I say that this plant is very tall and this plant is very short, what does that mean? At that time I am considering the height of plant. I am considering the height of plant. Means every character has two different forms. Means two different contrasting traits. In height we see tall and dwarf, long and short plant. Tall and dwarf are two different forms of a height. What side? Character. And tall and dwarf are two contrasting traits. Got it? Okay. This is suppose a trait tall, and every trait is represented by two alleles. Tall is represented by capital T, and dwarf is represented by small t. In monohybrid cross, what do? We made a cross between tall and dwarf. This tall capital T is crossed with small t, and we get capital T small t. This is the F1 hybrid. Here, this is again tall. F1 hybrid is what? Tall. We have taken two parents. One parent was tall and other is dwarf. The F1 generation, which is O-parent, that's tall. Can you tell me that what's the difference between F1 and this parent P1, tall? This is the tall parent, this is again tall parent. Is there any difference? Yes. What is the difference? The difference is that this parent tall is homozygous. Both the alleles are dominant alleles. Both the alleles are capital T, dominant alleles. In F1, we see two different types of alleles. One is capital T allele, tall allele, and second is small t, recessive allele. Two different alleles. When we see two different types of alleles are present, at that time we call it heterozygous. What do we call it? Heterozygous. What is heterozygous? What are heterozygous? Those organisms which are having two different alleles. Suppose capital T, both alleles are similar. Yeah, small t, both alleles are similar, so we call it homozygous. Now, proceed with the cross. This F1 individuals are self-crossed, means capital T is crossed with capital T small t. What we get? F2 generation, capital T, capital T small t, capital T, and small t. We get 4 individuals. This is the F2 generation. This is what? F2 generation. In F2 generation, beta, we get 4 offsprings. Out of these 4, 3 are tall, 1 is dwarf. Phenotypic ratio for monohybrid cross. How many individuals are tall? 1, 2, 3. Tall is to dwarf. How many are there? Dwarf. 3 are tall, 1 is dwarf. So ratio will be 3:1. The phenotypic ratio is 3:1. Now genotypic ratio. Genotypic ratio for monohybrid cross. We have 3 different types of genotypes. In F2 generation, we got 3 different types of genotypes: capital T, small t, and small t. Capital T, small t, small t. Capital T 1, capital T small t 2, and 1. So genotypic ratio is 1:2:1. This is all about monohybrid cross. Now we will talk about dihybrid cross. Dihybrid cross. What's dihybrid cross? Di means 2. In dihybrid cross, we study the inheritance of 2 characters. What is dihybrid cross? It involves the study of inheritance of 2 characters at a time. When we are studying 2 characters simultaneously, at that time we call it dihybrid cross. In dihybrid cross, we study the inheritance of, means the transmission of 2 characters. Why Mendel performed dihybrid cross? Here question arises that why Mendel performed dihybrid cross. Mendel performed the dihybrid cross to know the presence of any kind of interaction among characters during inheritance at a time of transmission of characters, when characters are transferred from parents to offsprings, at that time, does these characters interact with each other or not? So, why Mendel performed the dihybrid cross? To know the presence of interaction, to know the presence of interactions among characters during inheritance. During inheritance. Now, there are two possibilities. Two possibilities are there. Either these two characters, they interact with each other or they do not interact. So, two possibilities are there. Interaction present. There may be the interaction between these two characters. These two characters, they interact with each other at the time of inheritance. So, what will be the ratio? Does there any effect? Is there any effect on the ratio? Yes. If interaction is present between two genes, then ratio will depend on the type of interaction, on the what kind of interaction will occur between these two characters, and the ratio will depend on that. And second possibility is that there is no interaction, means interaction is absent. When there is no interaction, then the ratio will be assumed as 9:3:3:1. Got it? Now, suppose seed, in seed we take two characters. One is shape of the seed and color of the seed. Shape and color, these are two different characters of the seed, and every character has two different forms. For shape, it may be round or wrinkled. For color, it may be yellow or gray. Round shape is dominant over wrinkled. Yellow color is dominant over green. Round is represented by capital R. Wrinkled is represented by small r. Yellow, capital Y. Green, small r. We have two parents, parent 1, parent 2. We are studying the inheritance of two characters. So, one will be round yellow and second parent will be wrinkled green. Round yellow, capital R, capital Y. Small r, small y. So, capital R, capital Y, small r, small y. Round yellow parent is crossed with wrinkled green parent, and we get F1 hybrid. Capital R, small r, capital Y, small y. Round yellow F1 hybrid. Now, we have to find out the types of gametes. How many types of gametes are produced from the F1 hybrid? Because we have to perform the self cross. Now, what we are doing? Now, we have to do selfing. For selfing, we have to find out the different types of gametes. So types of gametes, to find out the type of gamete, we use the formula 2 raised to power n. What's n? N is number of heterozygotes. N is what? N is number of heterozygotes. In F1, how many heterozygotes are there? 2. Capital R small r 1, capital Y small y 2. So type of gametes 2 raised to power 2, that will be 4. 4 different types of gametes are produced from the F1 hybrid. Remember one point that we use 2 raised to power n formula to determine type of gamete, and this formula is applicable only on selfing. Whenever genes are linked with each other, at that time we cannot use this formula to find out the type of gamete, and this formula is only used when we are performing selfing. It is applicable only on selfing. Now, how many gametes are formed? 4 different types of gametes. To find out the type of gametes, we use fork line method. In this method, first we will take this character, 2 alleles, capital R, small r, capital R, small r. We take these 2 alleles, then this capital Y, small y, 2 times, capital Y, small y, capital Y, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. 4 different gametes are produced. Okay, see, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. 4 different types of gametes are produced from the F1 hybrid. Now we will perform the selfing. How many gametes are produced? 4. Capital R, capital Y 1, capital R, small y, small r, capital Y, small r, small y. These are the 4 different types of gametes which are produced. Now, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now we will cross this gamete with this, then this with this, then this with this, accordingly, then this with this. First capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Then capital R, capital R, capital Y, small y, capital R, small y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Now this capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, capital R, small r, small r, capital Y, small r, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y, small r, small r, capital Y, small y, small r, small r. This is how we can perform the dihybrid cross. How many different types of gametes are produced? 4. RY, round yellow. Now we have to find out the phenotypic ratio. Phenotypic ratio. What is phenotypic ratio? RY, round yellow. Round yellow, round green, round green, then wrinkled yellow, wrinkled yellow, wrinkled green. How many individuals are round yellow? Round yellow, round yellow, round yellow, round yellow, round yellow. This is round green. Round yellow, round green. Round yellow, round yellow, round yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. How many individuals are round yellow? 9. Round green, round green, round green, round green, round green. How many individuals are round green? 3. Now wrinkled yellow. Small or small, wrinkled. Wrinkled yellow, wrinkled yellow, wrinkled yellow. How many are wrinkled yellow? 3. Wrinkled green, 1. So ratio is 9:3:3:1. Now we'll check the genotypic ratio. Now genotypic ratio of diabetic class. There are very short cuts to find out the genotypic ratio of diabetic class. As you already seen in the monohybrid class, the genotypic ratio was 1:2:1. Monohybrid class into mono, mono into mono, di. In monohybrid class, the genotypic ratio is 1:2:1, 1:2:1, 1:1, 1, 1:2, 2, 1:1, 1. Now 2:1, 2, 2:2, 4, 2:1, 2. Now 1:1, 1, 1:2, 1, 1:1, 2, 4, 2, 1:1. This is the genotypic ratio of diabetic cross. Now, in diabetic cross, we take how many characters? 2, and we separate these 2 characters into 2 parts. We are taking what? Shape and color of the seed. We are taking shape and color of the seed. Okay, in shape, round, wrinkled, capital small, F1 hybrid. When F1 is self-crossed, we get capital R, capital R, capital small r, capital small r, small r, small r. Again here for color, yellow, green. This is F1 self cross, capital Y, capital Y, capital Y, small y, capital Y, small y, small y, small y. Now, what we have obtained from the monohybrid class? This is after generation. In after generation, one individual is always homozygous dominant and one individual is always homozygous recessive. One is homozygous dominant, one is homozygous recessive, and these 2 are heterozygous dominant. These 2 are heterozygous dominant. The ratio is 1:1. Same case here, 1:1. One homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive, as you already know it. Got it? Now, how we can find out the genotype of the dihybrid cross? We have separated the dihybrid cross into two parts, into two monohybrid crosses. Got it? First we take shape, then color, and out of these two characters we take one character horizontally and one character vertically. But how many times? 3 times, because we have 3 genotypes. So we take these two characters 3 times. How many times? 3 times. One character will be taken as on the x-axis and others taken on the y-axis. Suppose we take this shape horizontally on the x-axis. See, capital R, capital R, small r, small r, small r. How many times? 3 times. 1, capital R, capital R, capital R, small r, small r, small r, capital R, capital R, capital R, small r, small r, small r. We have taken this character horizontally. Now we will take this character vertically. Capital Y, capital Y, capital Y, small y, small y, small y, capital Y, capital Y, capital Y, small y, small y, small y, capital Y, capital Y, capital Y, small y, small y, small y. Got it? Now, how many integers are produced in the after generation of the monohybrid cross? 4, 1, 2, 3, 4. Out of 4, 1 is homozygous dominant, 1 is heterozygous, 1 is homozygous recessive, and 2 are heterozygous dominant. Capital R, it is homozygous dominant. How many times? 1. Color YY, dominant, homozygous dominant, 1. 1 into 1, 1. Capital R, small r, how many times? 2 times, because it's a heterozygous. 2 into YY, 1. 1 into 1, 1. Capital R, homozygous dominant, 1. Capital Y, small y, heterozygous dominant, 2. Capital R, small r, heterozygous, capital Y, small r, 2, 4. 1 into 2, 2. 1 into 1, 1. 2 into 1, 2. 1 into 1, 1. Got it? Now, remember these 3 points. Remember these. Homozygous dominant, always 1. Homozygous recessive, always 1. Heterozygous dominant, 2. ठीक? Heterozygous 2, homozygous 1. So I had this. What is the genotypic ratio? 1 to 1, 2, 4, 2, 1 to 1. So genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1. So genotypic ratio 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1. If we have to check whether we have done this cross rightly or wrongly, so for that we have to find out the total possible combinations. For total possible combinations, we use the formula 4 raised to power n. N is number of heterozygotes. N is what? N is number of heterozygotes. This formula is again applicable on your setting. And here, how many heterozygotes are there in dihybrid? 2. 4 into 4 is power 2 in 16. Here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Now we will check this genotypic ratio. If the sum of these genotypes is equal to 16, then it's right. If it's not equal to 16, then we have done some mistakes. Same here in phenotypic ratio. 9 plus 3, 12, 12 plus 3 15 plus 1 16, 1 plus 2 3 plus 1 4 plus 2 6 plus 4 10 plus 2 12 plus 1 13 plus 2 15 plus 1 16. So it means that we have done it rightly. Got it? Suppose if someone is asking you that what is the probability of capital R small r, capital Y small y. If someone is asking you what is the probability of capital R small r, capital Y small y, what will you do? There is no need to draw this long table. We can solve this within 2 seconds. How? Because we know that heterozygotes are 2, homozygotes are 1. It is hetero. It's hetero. 2 into 2, 4. The probability will be 4 by 16. Similarly, if someone is asking that, find out the probability of capital R, capital R, capital Y small y. Answer 1 into 2. Homozygotes are 1, heterozygotes are 2. Means 1 into 2, that's 2 by 16. 2 by 16 will be the probability, because in absolute generation of the radical class, total possible combinations is 16. Got it? Write down the question. Question is like this, that in F2 generation of the hybrid cross, in F2 generation of dihybrid cross, find out the probability of offsprings. Find out the probability of offsprings having, first of all, in dihybrid cross, we have to find out the probability of offsprings in F2 generation. Number 1, find out the probability of offsprings which are homozygous for both character, which are homozygous for both characters. Check here. We can use this method or second short. Homozygous for both capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, capital Y. 1 homo hetero, 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 homo, capital R, capital R, small y, small y. 2 small r, capital Y, capital Y. Then small r, small r, small y. 1 into 1 into 1 into 1. 1 plus 1 plus 1 plus 1. So answer will be 4 by 16. Second is heterozygous for both character. Heterozygous for both character. That is capital R small r, capital Y small y. 1, 2, 3, 4. So capital R small r, 2, capital Y small y. 2 into 2, 4. There is no need to draw this long table. So answer will be 4 by 16. Another question is that find out the probability of offsprings which are homozygous for one, homozygous for one character, and heterozygous for other character, which is homozygous for one character and heterozygous for other character. See, homo, this is homo hetero. Homo for 1, heterofather. Capital R, capital R, homozygous. Capital Y, small, heterozygous father. 1, hetero. This is homo for 1, heterofather. 2, homo for 1, heterofather. 3, hetero for 1, homofather. 4, homo for 1, heterofather. Homo for 1, heterofather. Small r, capital Y, small. How many there? 1, 2, 3, 4, 5, 6, 7, 8. So what will be the answer? Answer is 8 by 16. Capital R, capital R. Capital Y, small y. Capital R, capital R. Capital R, capital R. Capital Y, small y. Capital R, small r. Small y, small y. Capital R, small r. Capital Y, capital Y. Small r, capital Y, small y. 1, see, capital R, 1 into 2, 2, 2 into 1, 2, 2 into 1, 2, 1 into 2, 2 plus 2, 4, 4 plus 2, 6, plus 2 means 8. Answer 8 by 16. Like this. 1, 2, 3, 4, 5, 6, 7, 8. Find out the probability of the offsprings which are having parental phenotype. Parental phenotype. Where two parents, round yellow, wrinkled green. Round yellow, wrinkled green. Round yellow 9, wrinkled green 1. So answer will be 10, 10 by 16. How many individuals have parental genotype? Next question may be asked, that is parental genotype. That is capital R, capital R, capital Y, small r, small y, small y. That is 2 by 16. These two extremes are the parents. Rest individuals have recombinant or new genotypes. Only two individuals resemble with the parental genotype. So probability will be 2 by 16. Got it? Now write down the question. This is a very important question to be asked in various competitive exams like RGSO, NSEB or NEET. Write down the question. In a plant, tallness is dominant over dwarfness. Tallness is dominant over dwarfness, and red fruits are dominant over yellow fruits. If a plant with genotype capital T, small t, capital R, small r is crossed with the plant having genotype capital T, small r, small r, then find out the probability of offsprings. Very important questions, very important question to be asked in various competitive exams. How we can solve this question? In question, is saying that tallness is dominant over dwarfness. As you already know, tallness is dominant over dwarfness, and red fruits are dominant over yellow fruits. Means he is talking of dihybrid class, which read the two characters, tall, height, and color of the fruits. Okay, now what he is saying that if the plant, we have two plants, if the plant means plant 1 having the genotype capital T, small t, capital R, small r is crossed with capital T, small r plant, we have to find out the probability of offsprings which are having various phenotypes and genotypes. See, first we will find out the probability of offsprings which are tall and red. Tall and red. Okay. Now, capital T, capital R, small r is crossed with capital T, small r, small r. Okay. How many gametes are possible there? 4. Capital T, capital R, capital T, small r, small t, capital R, small t, small r. 4 gametes. Write down. Capital T, capital R, capital T, small r, small t, capital R, small t, small r. These are the 4 different types of gametes of this. Is crossed with capital T, small r, small r. How many gametes are formed there? Capital T, capital R, small t, small r. Capital T, small r, and small t, small r. So, capital T, capital R, small r. Capital T, small r, small r. Small t, capital T, small t, capital R, small r. Capital T, small r, small r. Capital T, small t, capital R, small r. Capital T, small r, small r. Small t, small r, small t, small r, small r. What do we have to find? Tall and red. Tall, red, 1. Tall, yellow. Tall, red. Tall, red. How many individuals are tall red? 3. So answer is 3. How many individuals are there total? In total, there are 8. 1, 2, 3, 4, 5, 6, 7, 8. So the probability of tall and red is 3 by 8. Is 3 by 8. Second, find out the probability of tall and dwarf. Tall and yellow. How many individuals are tall and yellow? Tall and yellow. Tall red, this is tall yellow. Tall yellow, and it's tall yellow. Again 3 by 8. 3 by 8. Now find out the probability of dwarf and red plants. Dwarf, small t, 1 red, that's 1 by 8. Find out the probability of offsprings or plants having dwarf and yellow phenotype, that is again 1 by 8. Got it? Okay. This is how we can solve these kind of questions. Write down one more question. question is like this: in a dihybrid cross, in a dihybrid cross, capital A, capital A, capital B, capital B, small a, small a. We are crossing capital A, capital A, capital B with small a, small a, small b, small b. The F2 progeny of capital A, capital A, capital B, capital B, capital A, capital A, capital B, capital B, capital A, small a, capital B, capital B, capital A, capital A, capital B, small b, and capital A, small a occur in the ratio of. We have to find out the ratio between these four integers. How can you solve this? This question can be solved within 10 seconds. See, in question, the four options are given there, like 1:2:4:2. Option B, 1:2:2:2. Option C, 2:1:1:2. Option D, 1:2:2:4. What will be the answer? See, first we will see this: capital A, capital A, capital B, capital B. As we already know that both the characters are in homozygous condition. 1 into 1, that will be 1. See, 1, 1, 1. Option C, this 2. So this option is removed. We'll eliminate this option. Option C is eliminated. Now there are three options: A, B, and D. Then comes to capital A, small a, capital B, capital B. Capital A, small a, heterozygous, 2. Capital B, homozygous, 2 in 1, 2. Now 2, 2, 2. Again same. No option is eliminated. Now we'll move to third. Capital A, capital B, small b. Homozygous of 1, it is 2. 1 and 2 means 2. C. 4. Option A eliminated. Option B and option D. Now there are two options, either B or D. Now we will move to fourth option. Capital A, small a, capital B, small b. Both are in heterozygous condition. 2 and 2, that is 4. Option 4, and this option is eliminated. So answer is option D. So we can solve this question within 10 seconds. 1 into 1, 1, 2 into 1, 2, 1 into 2, 2, 2 into 4. Within few seconds we can solve these kind of questions. Got it?